Greetings, ladies and gentlemen. My name is Dr. AJ Malat, and I am the co-founder and CEO of Ronawk. It is my pleasure to present to you here at the Biotech Showcase. Today, I'm going to share with you our company, Ronawk, and how we are accelerating next-generation therapies. At Ronawk, what we manufacture and what we produce is what we call the T-Block. And the T-Block is a revolutionary new cell culture platform. This platform is ultimately going to change how scientists, researchers, and producers culture cells going forward for high impact studies. T-Blocks reduce contamination risks by 92% when compared to standard culture vessels today. Let me say that again. T-Blocks reduce contamination risk by 92%. This is one of the biggest fears when it comes to cell production is the introduction of contamination. Because if cells become contaminated, a producer could actually lose their entire batch. So this reduction is significant. Furthermore, T-Blocks produce results 78% faster than other cell culture plastics. They are up to 82 times more efficient than standard cell culture plastics. And they reduce media consumption by 99% over standard cell culture plastics. Media is one of the most expensive components when it comes to producing cells, and the T-Blocks reduce that consumption. When you take all of these features and benefits together, T-Blocks ultimately reduce the cost of producing cells by 97% as compared to standard cell culture plastics. And to tell you more about this, I'm going to turn it over to my colleague, Scott.
Thank you, Dr. Malat. My name is Scott Lee. I'm the Chief Revenue Officer at Ronawk. What's really important here is when you're looking at total units and total media, you'll notice that we are going to reduce your material costs significantly. Then when you look at labor hours, that will be reduced. Even though we're reducing the number of hours and the number of units, you're actually going to get more cell growth out of our T-Blocks. Because we're reducing the number of vessels, because we're reducing the actual work someone has to do with cells, we're also bringing down the risk of contamination. Now, this is what the T-Block is. It is a bioprinted, synthetic, animal-free block. If you look at the picture in the top and the middle, that's the T-Block. And those little dots on there, those are the channels that the cells will grow in. What's great about this product too, is it supersizes all traditional models that are out there. Whether you grow in a dish in the lower right corner, a 6-well plate in the top left, or a 12-well plate, or even roller bottles, cell factories, bioreactors, this little block can help maximize the cell growth in all of those vessels. In the lower left, you'll actually see a magnified picture off of a microscope showing you our core channel and where the cells grow. On the next slide, we're going to show you how easy it is for any lab anywhere in the world to adopt this program. First, they would marinate the block just like they currently do. Pick your marinade. Whether it be fibronectin, gelatin, these are common practices among labs now. Step 2, they will add their cell of choice, whether it's 293 cell, whether it's a HeLa cell, or a primary cell from animal or human. They will add this just like they currently do. Step 3, they will change out the media, significantly less media than they're currently using. We want to keep those cells fed every couple days. Now, here's where it gets really interesting. In step 4, instead of subculturing, which is a process of taking the cells out, chemical work with them, spinning them down, replating them, all we're going to do is add a second, third, fourth block and keep that cell growing. Now, what's great about this stage 2, we could take block 1 and harvest the cells, but keep the culture growing in block 2, block 3, block 4, or we just start with a lot of blocks and let it go. That's where we limit contamination significantly because all it is adding a new block, which takes seconds. In step 5, this is a great comparison because ultimately a lot of people are going to want to harvest these cells. What you see in this chart is a traditional 2D method versus our block at 15 days, 30 days, and 60 days. What's significant here is we are not speeding up how cells split. What we are doing is giving them unfettered ability to do so. So they're expanding across the X, Y, and Z axis inside of our block. So that's where you get that exponential growth. And even more exciting is what we've got coming in 2022. This is an incredibly elastic and expandable platform. Our two next blocks are going to be an X block. This is really cool for all the scientists out there, because with our proprietary reagent, we're going to be able to add that to the block, and the block just dissolves and you're left with a ton of cells. We're seeing cell extraction of 90 to 95%, which is outstanding. The next block we're going to add takes this 5-step process down to a 4-step process, and that's the bioactive block. It does not need to be coded. This means for virtually every cell application out there, they can just add the cells. They don't need to code the block, and that creates a huge advantage going forward. Let's create some visuals for you to see what this is actually reducing in the lab. So we put the T block next to a quarter to give you an idea of how small that is. One block replaces 120 T75s. A T75 is the most common growth vessel anywhere in the world. The next group is 5-liter bioreactor. You will find 5-liter bioreactors in every biopharma, every contract research organization, and every CDMO throughout the world. 2 of our blocks replace 2 5-liter bioreactors. Or in another way to think of it, putting our blocks in those bioreactors supersizes them. On the last picture, because you can fit 120 T75s inside of a CO2 incubator, 5 of our blocks replace the production of 5 CO2 incubators. So when you're thinking about equipment and material costs, that's where you're really just cutting your costs significantly, and you're able to do far more with far less. Now, what also makes this block really neat for investors and biopharma is this is an FDA 510(k) exempt Class 1 device. This means we're able to enter markets much faster. We are not reinventing the wheel. We have just reshaped it and supersized it. We're also protected under a lot of trade secrets and patents pending. So this is a very unique thing that people can just not copy. You've heard me talk about other cell culture vessels, but what the T block allows to do is more than any one of these single vessels by themselves will do. So not only have we given the researcher, the scientist, the pharmaceutical company a platform to expand on, we've given them a tool that does far more than their existing tools do and maximizes the space in those tools. I'm going to turn this back over to Dr. Milan, and thank you for your time.
Thanks, Scott. Now, we've been telling you about the features that we're excited about, but one of the things I absolutely enjoy is the fact that it's what our customers are saying about our blocks. If you look here, just to highlight 2 of their quotes, one has talked about what we've told you today. These T blocks eliminate the need to subculture cells, which reduces contamination risks and can even help increase the number of quality cells that are produced. Furthermore, on the medical side, looking downstream, the T blocks are the starting point at the bench for allowing researchers to accelerate new therapies. But these therapies have significant implications at the bedside, and physicians are already starting to recognize that these T blocks could improve the outcomes for patients that have undergone reconstructive surgeries, cancer removal, burns, and other traumas to the body. I'm now going to turn it over to my colleague Tom to tell you a little bit more about the business.
Hi, this is Tom Jantz, and I'm the president and COO for Ronawk. I want to talk about how T blocks impact our customers and the markets that T blocks are applicable to. T blocks are a powerful tool that allows scientists to accelerate their research into next generation therapies. It does this by streamlining operational efficiencies, reducing development and manufacturing costs, and ultimately shortening time to market. On slide 10, it states that all biology starts with cells. The need to grow more cells is a major bottleneck. As Scott explained, Ronawk removes this bottleneck. While we start in the $19 billion cell culture market, T-Blocks also enhance the production of biologics, a $325 billion market, which then leads to the organoid market, an $800 billion market, and ultimately the personalized medicine market, including regenerative and precision medicine, a $1.9 trillion market. The T-Block is the platform that enables all these markets to rapidly accelerate. The markets described on the previous slide are large, and the flow of business is somewhat established. Ronawk has received purchase orders from universities and research institutes, and is currently in conversations with several CROs and CDMOs for the purchase of T-Blocks and/or new products. This interest is being driven by the unique value proposition that Ronawk provides. That value is a shorter time to market, which ultimately makes our customers more competitive. We are busy and have many opportunities in front of us, but are currently open to equity-based or other financing discussions that will allow Ronawk to explore expanding market presence, increasing sales penetration, securing additional IP for synergistic products, and launching additional products into new markets.
And thank you for that, Tom. Now I want to tell you just a little bit about our team, which I'm very proud of. At our core, the four of us have been working together in some capacity or another for the past decade. Together, we have a combined experience of 75 years. And even more so, the fact that we have pulled everyone together, we have created a unique balance of both scientific expertise with business experience. As the co-founder and CEO, I am the original inventor of the T-Blocks. I've spent the past 12 years of my career focusing on tissue engineering and manipulating cells to accelerate their production for downstream applications. My co-founder and our Chief Technology Officer, Heather Decker, is the one that has been driving the development of a variety of applications for the T-Blocks. She is an expert in analytical imaging, and she even ran her own lab for quite a while. Tom Jantz, our president and COO, has come to us with over 25 years of financial and operational experience. We rely on his expertise in startups as he has exited multiple companies, and in 2020 was named the CFO of the year in Kansas City. And Scott Lee, our Chief Revenue Officer, additionally brings 25 years of experience selling in the cell culture market. He has sold a variety of products that our competitors have used and our customers know. But when Scott joined our team, he was most excited about how we could change that market. And we've relied on him to make introductions through his network, which has gotten scientists excited, as well as our potential end users here. So with that, I'd like to thank you for your time. I look forward to your questions and inquiries, and am excited to tell you more about Ronawk. Thank you very much.